Let's go on with DNA replication exam questions, right? So the first question here, choose the relationship which is true regarding the central dogma of molecular biology. Now, the central dogma of molecular biology simply tells us that DNA can change. Remember our cells, for eukaryotic cells, our cells have got what? Have got a nucleus, right? So in that nucleus, we actually find DNA. But think of prokaryotic cells like bacteria, they don't have a nucleus, so DNA is found as a nucleoid. DNA is just together like that in a structure known as a nucleoid. There is no nucleus. Okay. So we are going to have DNA. Before, let's say I've cut my hair, I need to form hair. That is protein, right? Hair is basically made of proteins. So those proteins are going to, the instruction to form them will come from DNA. But before DNA can form those proteins, it has to be converted to RNA, specifically mRNA. And then RNA now will be converted to proteins. So the first stage, but before we can actually even convert DNA to RNA, we can get DNA and make a copy of DNA. All right. So these are the phases of what we call the central dogma of biology. DNA making a copy is known as replication. And then DNA going into RNA, that is known as transcription. Now DNA going into proteins, that is known as translation. So these are the phases of molecular biology. So it is translation, replication, proteins. Is that true? Is it replication that form proteins? No. Transcription, translation, then proteins. This is true, right? Because after translation, we have got proteins. Transcription, protein, no. Transcription, protein, no. Transcription, translation, then replication, no. Because replication is supposed to be the first one. Okay, so that is it for this one. Semi-conservative replication of DNA was first demonstrated in. Now, semi-conservative replication was first demonstrated in Escherichia coli or E. coli. Please, if you can't remember, go and consider the tutorial on DNA replication. It was fully explained the entire topic. Correct sequence of stages of prokaryotic DNA replication is. Now, the stages of replication, the first stage is always initiation. All the three stages of central dogma of molecular biology always starts with initiation. After initiation, then we are going to have elongation. Elongation is also known as polymerization because we are forming a chain. So polymerization. And then after that, then we are going to have termination. Like we have already formed our chain, so we need to terminate or to stop it. Okay. And then after that, we are now going to have modifications. That will be the lasting modification. This modification, if it is replication, if it is transcription, we are going to call them post-transcriptional modifications. If it is translation, post-translational modifications. So which one is going to be the answer? It is initiation, that is the first thing, polymerization, and then termination. Okay, so that is option 2. Choose the relationship which is true regarding the central dogma of molecular biology. DNA. So the first one they've mentioned here is transcription. It's DNA to RNA. Well, you know that is true, right? But translation is not RNA to DNA. So this is wrong. The second option, DNA to DNA replication, that is true, right? DNA to RNA, that is transcription, that is true, right? RNA to proteins, that is translation. This is true. All right. So of course we already know that the others are not going to be true, but let's disqualify them. RNA to DNA, that is not transcription, so that one is not correct. And then RNA to RNA, that's not replication. DNA to RNA, that is not transcription. During DNA replication, nucleotides are added to dash. Now in replication, you always use DNA has got a strand. You have got one that is running from the 3 prime end to the 5 prime end, and then the other one running from the 5 prime end to the 3 prime end. So the chain that we use in replication is this one that is running from the 3 prime to the 5 prime, meaning that the guy that we are forming is going to be running in the 5 prime to 3 prime direction. Okay, so the guy that will be not 3 prime to 5 prime, but the guy we are going to be forming, or the new strand we are forming, is going to be running from 5 prime to 3 prime direction. Okay, so this is the strand that we are going to be forming. So meaning that if I've got, let's say, DNA molecule, so it is like this. Now we know that this is the 5 prime end, this is the 3 prime end, and we are saying we're making the new chain from 5 prime to 3 prime. So where do you think we are adding things? We are basically adding things here. So we are going to add something here. What's that end? That's the 3 prime end. So during DNA replication, nucleotides are added to the 3 prime end of the DNA primer. Okay, DNA primer. But the first thing that we do is that we are going to lay down an RNA primer. That is the first thing that we do. The primers that we lay down are laid by the enzyme known as the primase, and that is an RNA molecule. So it will actually be by RNA to, we are going to be adding nucleotides to RNA primer. Okay, that's the first thing that we do. But then to continue the process, we are going to be adding to the one end, and that one end has got DNA. Hope we understand this very well. The first primer that we lay down, led by primase, is going to be RNA, but the others to come are going to be DNA primers. To lay the primer that we laid down is coming from RNA, so the answer there is going to be to RNA. That is the primer. And then what to continue, we'll just be adding to the new strand that we are forming. Stages of DNA replication can be represented by the following order: initiation, separation of daughters. So when you have got the strands, here are the two strands. Okay, so helicase is going to come and it will start separating these two strands, but that is after initiation has already happened, because initiation is actually the attachment of what we call DNA, or the initiation factor, to the point where replication is going to start, and we call that as a replication, the origin of replication. So initiation, separation of DNA strand, proofreading, elongation. Proofreading is modification. That thing comes at the end, is modification, yeah. And then two, separation of data strand, initiation, elongation, proofreading. I'm going to put a dot on this one. Option 3, initiation, separation of data strand, elongation, and proofreading. I'll also put a dot, elongation, initiation, but this one is disorganized. You can't start with elongation then initiation. Initiation, elongation, proofreading, separation of strand. You can't separate a strand after you've done everything. So the first thing is we are going to initiate the process, right? Initiation is a first, and then we separate the strand. As you are going to be separating the strand, that's when elongation is going to be happening. Okay, so we're supposed to start with initiation. Option 3, that is the one. Choose a statement which is false. After replication, each daughter DNA contains one odd strand and one new strand. So what happens is this. Let's say we started with this structure. After replication, these two are going to separate like this. And what will happen is that to each one of these guys, we're going to add a new strand. And then this is the first replication that has happened. When we're having the second replication, What is going to happen is that again these strands are going to separate. Can you see that we have got two red ones? Those red ones are going to separate, right? Like that. And then we have got two blue ones. They are also going to separate. But remember, the new strand that we are adding after replication is blue, isn't it? So this one, we are going to add a blue strand, a blue strand, a blue strand, a blue strand. So you can even work out this in terms of percentages. You can see that after first division, you have got 50% odd and 50% new. In other words, you only have hybrid. So it is 100% hybrid. Hybrid means combination of odd and new. But after a second replication, you have got these two are hybrids. They have got new and odd. So 50% hybrid and then 50% new. But of the hybrids, can you see that you have got one new strand and new one odd strand? In other words, you have got 25% of the hybrid being odd, 25% of the hybrid being new. So altogether, the new strand is going to be this 25, which the odd strands will just be 25%. The new strand will be 25% in the hybrid and 25 and 50% which is alone. That is 75. Hope you get the math right. After replication, each daughter DNA contains one odd strand and one new strand. Is that true? After replication, each daughter DNA, each, this is true, isn't it? One odd and one new. In their experiment, Michelson and Starr used E. coli. This is also true. We even mentioned that at the beginning. Michelson and Starr initially grew E. coli cells in a medium containing nitrogen 15 isotope for many generations. This is true. This red will actually be representing nitrogen 15. And then the blue, the new, that's a new medium in which we are growing the E. coli, is representing nitrogen 14. Michelson and Starr initially grew E. coli in medium containing nitrogen 14 isotope for many generations. That's not true. That was a second, not initially. Michelson and Starr analyzed DNA samples using cesium chloride density gradient equilibrium centrifugation. This is true. What happens is that they now got these E. coli, put them in test tubes, and then spinned the E. coli that sorted at the very bottom. They knew that was the heavier one. It had nitrogen 15. The one on top most had nitrogen 14. So that's how they had to tell. You can still follow that to understand on our class on replication. Choose a molecule which opens the DNA, creating a replication fork. So what forms the replication fork is DNA helicase, right? So look at this. This is your DNA molecule that you are separating. There are going to be some important enzymes. Now you know that before you can have this, DNA is like this. It's coiled before it can become straight. So the enzymes that are doing work here, the first one is DNA gyrase. What is gyrase doing? Gyrase is there to remove supercoils. Okay, it's removing supercoils to turn it from being like this to being straight so that it can be easy to separate. And then helicase is going to come in. What is helicase doing? It is separating the two strands. Now the strands which have been separated, you are going to have another protein here known as single stranded binding protein, SSB protein. That one is binding to these regions that have been separated to prevent them from coming back together. And then as helicase is going forward, what will happen is that DNA polymerase is going to be forming a new strand like this. For the strand running 3' to 5', it will be going forward. For the strand running 5' to 3', it will be going behind. Because the new strand can only be formed in the 5' to 3' direction. That is why, that is because DNA polymerase 2 can only go from 3' to 5'. So the new strand can only be formed from 5' to 3'. Hope you get that. Okay. And then for this strand that is running from 5 prime to 3 prime, what will happen is that first, because at the beginning we first laid down a primer before DNA polymerase can come in. A primer is going to be laid. A primer is basically a nucleotide, right? Few groups of nucleotides. They are going to be laid down by what we call RNA primase. Primase, it's RNA molecule. It will lay down a primer, and then DNA polymerase can continue. But on this, so this strand is known as the leading strand, which is running from 3 prime to 5 prime. The one which is running from 5 prime to 3 prime, this one is known as the lagging strand, because for this one you are going to have RNA polymerase working in the opposite direction, because RNA polymerase can't read from 5 prime to 3 prime, so it will be working in the opposite direction. That's why RNA polymerase is a bi-directional enzyme. It will be working forward and back. Okay. But for the lagging strand, the primase is going to lay down a lot of primers. These polymers, we they are now going to the DNA polymerase is going to now come and form short strands, short strands, and those are known as Okazaki fragments. Okazaki fragments. And then what will come and complete joining these, forming the phosphodiester bonds, is going to be DNA ligase. Okay, ligase. So take note. DNA gyrase is also known as topoisomerase. Okay, so I've explained what each one of these enzymes here are doing. At the end of the lagging strand, DNA synthesis, phosphodiester bonds between adjacent nucleotides break. They don't break, they are supposed to form. Bonds between the nitrogen base and deoxyribose sugar breaks. No bond breaks, we are forming bonds. The leading strand produces Okazaki. No, it's the lagging strand. Hydrogen bonds between the nucleotides of the two strands break. No, we don't break. Okay. DNA ligase catalyzes the sealing of the gap between adjacent DNA fragments. And this is DNA ligase catalyzes the sealing of gaps between adjacent DNA fragments. This is true among the Okazaki fragments, right? That's what we said. Bacteria cells grown in heavy nitrogen isotope and allowed to replicate 3 times in a culture of light nitrogen isotope will show the following results. Let me do it again, though I already showed. Let me even write for you the key. Nitrogen 15, I'm going to represent it by, let me say, by blue. Okay, I'll represent it by blue. And nitrogen 15 is what we start with in this experiment done by Nick Nicholson and Starr. And then nitrogen 14 are represented by red. So initially, the entire DNA, we grew it in an environment with nitrogen 15. So it has all been completely labeled with a blue color, meaning that it has got nitrogen 15. After the first replication, what happened is that the two strands had to separate. You can see they have separated. For each one of these strands, because now we are growing our bacteria in nitrogen 14, a new strand is going to have nitrogen 14. And then after that, we are going to do a second replication. For a second replication, all these strands will separate. You can see the red ones there, and then the blue ones have also separated. Let me put it here, and then another one here. But because we are still growing our bacteria in nitrogen 14, each strand is going to carry nitrogen 14 as a new strand. So it will look like that. And then after the third replication, what is going to happen? All these strands will separate. There are 1, 2, 3, 4, 5, 6, 7, 8. So 8 strands. Of the 8, 6 are red. So the 6 red, I'll put 1, 2, 3, 4, 5. Let me put the sixth one here. 6. 1, 2, 3, 4, 5, 6. And then we have got 2 blue ones. But because another replication is happening in an environment with nitrogen 14, each one of these strands is going to carry a red strand. each one of them. So now what can you notice? So remember, this is after the third replication, and that's the question we have. How many red strands can you see? And remember, in each one of these strands, you have got two molecules, right? So you have got two, so 2, 4, 6, 8, 10, 12, and then 13, 14. So you have got 14 strands which are what? Which are red. And it's out of what? How many in total do you have? If you count these 1, 2, 3, 4, 5, 6, 7, 8, there are 8, but in each one of them there are 2. So 8 times 2 is 16 out of 16. The blue one is only 2 out of 16. So what you are simply doing here is converting this one into a percentage. What are we going to have here as a percentage? Okay, 2 out of 16, that is a 12.5%, and then 14 out of 16, that is 87.5%. Now remember, this is a new strand, percentage of new strand. This is a percentage of old strand. But then again, remember, of these, can you see that two strands, this one and this one, they're hybrids? You get that, right? So we have got 2 out of 16, which are what? Hybrid. And remember, 2 out of 16 is 12.5%. These guys are hybrid. And then we have got how many? Oh, sorry, not out of 16, because I'm counting the, this has 1 and this one has 1. So it is 2 out of 8, not 16 now, because I'm not counting individual. So it is 2 out of 8. And this is 25%. This is a hybrid. And then we have got these 1, 2, 3, 4, 5, 6. So 6 out of 8. That is a what? A 75%. These are what? These are new strands. Okay, they are not hybrids, completely new. Okay. Or they have got light nitrogen alone because they are red. But these guys have got a combination. These 25% to be having a combination of heavy and light. So let's answer. So what to be the results? 100% heavy DNA? No. 100% light? No. 50% hybrid? 50% heavy? No. That will be after the second replication, not the third. 25% hybrid and 75% light. Is that true? Yes. 25% light, 25% heavy, and 50% hybrid. This is not true. Right. So this is how you answer this question. During replication, new nucleotides are added to the, so remember we had already mentioned. So for the growing strand, this is a phosphate. So new nucleotides are being added to here. This is the 3' end, because where I've got here, this is the 5' end. The 3' end has got an OH. Okay. So OH group of 3' end of deoxyribose. That's DNA. Because ribose is found in RNA, but we are adding to DNA. Okay. So that is it. The stages of DNA replication can be represented by the following in reverse order. Remember, this is reverse. So if it is reverse, we'll not start with initiation. Even this one is out, even this one is out, right? Separation of daughter DNA, proofreading, elongation, initiation. But if you look at option, anyway, let's go to for elongation, initiation, separation of daughter strands, proofreading. So this one is a bit interesting. So remember, we're supposed to start with what is the first stage? Initiation. After initiation, elongation. After elongation, then termination. After termination, modifications. That's why you can also have the proofreading. But the only thing is this: proofreading, controlling mistakes doesn't just happen after termination. You can actually control the mistakes before you can terminate the chain, like when you are just synthesizing the chain. Okay? So separation of daughter DNA, proofreading, elongation, initiation. Okay, let me pause that. But this one, elongation, that's supposed to initiation, separation of the daughter strands of the daughter DNA, and then proofreading. Now, you don't do proofreading after you have removed the chain. Actually, just before you can, what we are saying is this is the old chain that you have, and then this is a new chain that you are making. So even before you can remove this new chain that you are making, you can do proofreading, right? You can do proofreading. But now what we are doing here is the daughter strand is a strand that you are forming. The parent is the one from where the one that you are forming is coming from. So before you can separate this one, proofreading is actually going to happen. Okay? So before separation. Because after termination, then there's going to be separation. But before separation can happen, you are going to have proofreading. Okay, hope we get that. So meaning that we're supposed to start with remember we are going from the back. So let me just clarify, make sure that this thing is correctly mentioned. Proofreading is correcting mistakes. They can happen in elongation. They can happen after you terminate the chain. They can basically happen anytime. Okay, they can happen anytime. Let me correct that well. So after termination or before termination, corrections can happen. So meaning that proofreading can be the separation and proofreading can swap. So meaning that we are going to start with separation of the DNA strand, then proofreading, meaning that we are making corrections before separation of the strand. Remember, we're going in the opposite direction, and then elongation, then initiation. But for we are starting with elongation, then initiation. These two steps are supposed to be at the end of elongation, so that's why this one is not correct. At the end of the lagging strand, DNA synthesis, phosphodiester bonds between adjacent nucleotides break, no. Bonds, no. Bonds between nitrogen base and deox break, no. A number of Okazaki fragments are produced. This is true, right? Hydrogen bonds. Every time you hear break, that's not true. Helicates characterize the seeding. It's not helicates, it's ligase. Which reaction in DNA replication is catalyzed by DNA ligase? Remember, it is the formation of phosphodiester bonds in the lagging strand. Okay, addition of new nucleotides to the lagging strand. Let me put a dot. Addition of new nucleotides in the leading, not leading. Base pairing of the template of the newly formed DNA, not really. Formation of phosphodiester bonds between 3' hydroxide of one Okazaki fragment and the 5' phosphate of the next on the lagging strand. This is the thing. Synthesis of the short RNA primer, not really. So why was the first one wrong? Addition of new nucleotides. We don't really add nucleotides. We are just forming phosphodiester bonds of what the nucleotides which are already there. During DNA replication, separation of DNA strands require, remember it's separation, right? Hydrolysis of phosphodiester bonds, DNA ligase, hydrolysis of GC and AT complementary base pairing, topoisomerase activity. Okay. Separation of DNA strands. What do we require? Look at this. You have got these two strands. What caused this first separation? It is helicase, isn't it? But what is helicase doing? Helicase will start breaking down the bonds between cytosine and guanine and adenine and thymine. So it is hydrolysis of guanine and cytosine and then adenine and thymine. Choose the enzyme which is correctly matched with its function. Topoisomerase causes the initial strand separation of DNA. No. DNA remove supercoils. No. What to remove supercoils is what? So if it is, let's say, matching, let's do matching. Let's start with the causes in initial strand separation. This is DNA. DNA will be the one causing the first separation. Topoisomerase is the removal of supercoils. And then helicase produces RNA. No, separates two DNA strands. This is a function for helicase. And then primase produces RNA primer. This is its function. Single-stranded prevents separated DNA from rejoining. So that's the correct one. Which reaction in DNA replication is catalyzed by ligase? So we've already answered this one. What did we say? We said 4. Elongation of the leading strand during DNA synthesis progresses away from the replication fork. Now, when you are making, what we form is something like this, right? Helicase is the one that has formed the replication fork. So where helicase is going, this is what we call the direction of the replication fork. So when you are forming a strand, it goes in the direction of the replication fork. So this one is not true. Occurs in the 3' to 5' direction. Elongation. Elongation is going to read the chain running from 3' to 5'. That is how we are going to be going. But let me put a dot. But the new strand, so you need to be careful for this one. If they're talking about the new strand, it will be formed in the 5' to 3'. We're talking about the strand that we are looking at. We are going from 3' to 5'. Produces Okazaki fragments, leading, no. Uses a DNA primer. The elongation is not elongation, that's initiation. Depends on the action of DNA polymerase. Yes. Okay. So this one is not going to be true because we are talking about elongation. Elongation, the new chain that we are forming will be going from 5' to 3'. But the chain we are using is going from 3' to 5'. So please be very careful with this. Bacteria such as E. coli grow on nitrogen 15 medium, are transferred to nitrogen 14 medium and allowed to grow for one generation. DNA extracted from this bacteria is centrifuged. What density distribution? Now remember, this is only one generation, and for one generation we've been talking about this. We have got 50% hybrid. You can even draw it yourself, okay? And then 50% new. But remember the hybrid, we have got 50% which is old and 50% which is new. So one high density and one low density DNA, really not. One high density and one hybrid. High density means nitrogen 15, no. This is not true. Only hybrid, no. Half light and half hybrid. This is true, isn't it? Because 50% hybrid, 50% light, which is the new one. Initiation of DNA replication takes place at the, it is going to take place at what we call the origin of replication after DNA binds, and that's what causes the initial separation before helicase can take off. Enzyme or enzymes and protein or proteins involved in denaturation of DNA replication are helicase and single-stranded binding protein, topoisomerase. Remember denaturation, you have got this double-stranded guy, but you are trying to separate it so that it can become like this. So helicase is playing a role. Single-stranded binding protein, of course, is also preventing the coming back together, but let me put a dot. Topoisomerase, really not in denaturation. RNA polymerase, not in denaturation. DNA polymerase, not in denaturation. Helicase, single-stranded binding protein, and DNA polymerase. This is true. DNA polymerase is going to be going forward on the separated strand, and that new strand that is forming is actually also preventing the coming together, because as this guy is moving, it's actually also getting to help in that process. Okay, so if you come to, anyway, let me just remove DNA polymerase. When you come to RNA polymerase in transcription is the one which will be separating the strand and adding new strands or making a new strand. But DNA polymerase will just be making a new strand, not really being involved in the denaturation. So the best option here would actually be one. If it was RNA polymerase also causes the breakdown of the chain, but that is transcription. Choose a statement which is correct. The leading strand grows in the opposite direction. No, it's the lagging strand which does. The overall growth of all the Okazaki fragments is opposite to the direction of the replication fork. This is true. Okay. Individual Okazaki fragments grow in the opposite direction to the replication fork. Really not. The individual ones, they are being laid down by primase, and primase can put them in the forward direction. The leading DNA strand is synthesized in the 3' to 5'. No, we are reading the strand from 3' to 5', but not the new strand being synthesized in 3' to 5'. Okazaki fragment is synthesized in the 3' to 5'. Really not. What it will be synthesized in the 3' to 5' is going to be the joining. Okay, when you are joining these Okazaki fragments, not the Okazaki fragments themselves, really. Okay, let's do this. Okazaki fragments are formed by what? It's DNA polymerase, isn't it? DNA polymerase can only read something that is running from 3' to 5'. The meaning of that is that it can only form something in the 3', 5' to 3' direction. So you get it why this one can be the answer. Okay, so that is it. If the sequence of one strand of DNA is, so you can see, this is DNA. We have got 5' end, T, C, G, A, T, C, 3' end. What will be the complementary strand? Remember 5' will be 3', T pairs up with A, C with G, G with C, A with T, T with A, C with G, and then 5' end. So which one do you think is gonna be the answer here? 3' A, G, C, T, A, G, 5'. So this one. DNA polymerase the 3 adds nucleotide to the 5' end of the RNA primer. Do we add to the 5'? No. 3' end of RNA primer. Okay, this can be true. 3' end in the place of primer RNA that it is removed on a single stranded template without need for RNA primer to both ends of the RNA. Okay, so the best is that one, 3' end of the prim. Enzymatic ligase catalyze the formation of covalent bonds between dash of the nucleotide and dash of the adjacent nucleotide. So remember, this is ligase forming bonds. It is always the 3' end hydroxyl. The 5' end has got a phosphate, not hydroxyl. The 3' end has got the hydroxyl. Okay, so 3' end hydroxy and 5' phosphate. The enzyme removes the supercoils in the DNA helices in front of the replication fork while separating double-stranded DNA into a single strand. What removes the supercoil? So the enzyme dash removes supercoils. What removes the supercoils is helices, right? In the DNA helices in front of the replication fork, while dash separates DNA stranded into a single stranded. Okay, supercoils is not helices, it's topoisomerase. And then what is it? Removing the double strand, separating it is now helices. Each Okazaki fragment is synthesized in the, remember each is going to be synthesized because it's Okazaki fragments are made by DNA polymerase. DNA polymerase can only synthesize in the 5' to 3', right? Each. Okay. The origin of the replication fork is reaching. So the replication fork, remember adenine and thymine has got 2 double bonds. Cytosine and guanine has got 3 double bonds. So which one do you think is easier to separate? So then in a thymine, that's why the replication fork is reaching these two guys. During DNA replication, an RNA primer is required for leading strand synthesis and not for lagging. It's actually required for both. Required for leading strand synthesis and DNA primer for lagging strand. That's not true. Added to the 3' end of Okazaki fragment requires for both leading and lagging strand. This is true. Synthesized by enzyme DNA polymerase, really not. Added to the 3' end of Okazaki fragment. Now we're not adding the primers to the Okazaki fragment. We are first laying down the primer, and then the primers will be connected to form Okazaki fragments. Okay. Choose the relationship which is true regarding central dogma. DNA to RNA transcription. RNA to DNA, this is not true. DNA to DNA replication, DNA to RNA transcription, and then protein, this is true. So these others won't be true. Which of the following is correct? DNA is a template only for DNA replication. Is that true? It's not only. Also for transcription, we are going to be using DNA. DNA is a template for synthesis of all RNA types. Is that true? Yes. Because all RNA comes from DNA. Translation takes place before transcription. No, it is after. RNA is a template for DNA transcription. RNA, template for transcription. No, transcription, you are going to remember this is DNA transcription. You start with DNA and then you are going to RNA. DNA replication takes place after translation. No, it's before. The mode of DNA replication in E. coli is, remember, in E. coli is going to be non-conservative, and then it is bidirectional. Actually, semi-conservative. You have got a new strand being there, and then the old strand coming in. And then it happens both in the 5' to 3' and the 3' to 5'. DNA replication starts when the phosphodiester bonds between adjacent nucleotides break. Okay, replication, of course. But you don't break phosphodiester bonds. What you break are hydrogen bonds between adenine and thymine and cytosine and guanine. Bonds between nitrogenous bases and the deoxyribose breaks. No. Look at this. Here is a phosphate. Here is a pentose sugar. Here is a base. It can be adenine, thymine, or anything. Okay. And then you can have another base here, like this. And then another base. So this side as well going to have another base. Even this side another base. So what are the bonds you can see here? This bond between a phosphate and a pentose sugar, this is a pentose sugar, this is a base. This is known as a phosphate ester bond. Now, when you are connecting two nucleotides, can you see that you have got a phosphate with two pentose sugars? So the bond becomes known as phosphodiester bond. So phosphodiester bond is between the two guys. And then you have got this bond between the nitrogenous base and the pentose sugar. This is known as N glycosidic bond. What is going to be separated are the hydrogen bonds, 2 between adenine and thymine and 3 between guanine and cytosine. These are hydrogen bonds. These are the ones which are separated when replication is happening. So not between the base and the deoxyribose. Leading strand produces short segments. What produces short segments is the lagging strand. Hydrogen bonds between the nucleotides of its two strands break. Yes. DNA polymerase. Short strands of RNA. No, that's not the bonds which are broken. What is the role of topoisomerase in replication of DNA? It binds to the origin of replication site within the double-stranded. Not really. It unzips the double strand. No. What will unzip is helicase or DNA. Relaxes double-stranded DNA in readiness for smooth replication. That is the thing. Relaxes, removes the supercoils, joins the DNA nucleotides together. No. It is a single-stranded DNA stabilizer. No. Choose the molecule which opens double-stranded DNA to create a replication fork. You know, right? DNA helicase. At the end of the lagging strand, DNA synthesis, phosphodiester bonds between nucleotides break, not breaking. Everything to do with break, you remove them, right? Gaps between adjacent DNA fragments are sealed at the end of the lagging strand. We said this is true. This is actually repetition bonds. The leading strand produces Okazaki. No. DNA during replication of DNA, only DNA primer is produced during the lagging strand. No. Many RNA. Okay, this is only one DNA primer is produced during the leading strand. Only one DNA primer. It's not a DNA primer, it's an RNA primer. If it was RNA, the answer would have been correct. Many RNA primers are produced during the lagging strand. That is correct. Lagging strand, a lot of RNA primers. Only one RNA primer is produced during the lagging? No. Only one Okazaki fragment is produced during the leading. Leading has nothing to do with Okazaki fragments. Many DNA primers are produced during the lagging. They are not DNA fragments, supposed to be RNA. Which of the following enzymes is used to join fragments of DNA? Remember, its fragments, it's DNA polymerase. Okay, but wait, this is actually fragments. It's DNA ligase, so watch out, right? It's ligase fragments. The diagram given below shows the process of DNA replication at the replication fork. Study it and then answer the questions that follow. You can see I'm moving from 3 prime to 5 prime and then like that. So we have got D, we have got B and C there. The strand labeled B is there. B, look at B. Can you see that we are forming it from on the strand from 3 prime to 5 prime? That is the leading strand. Even an arrow is showing that you are going forward, you are going in the direction of the replication fork. That is the leading strand. Which of the following contains the genetic information for the synthesis of one specific protein? Remember, genetic information is found where? Which of the following contains genetic information for the synthesis of one specific protein? Messenger RNA, transfer RNA, ribosomal RNA, messenger RNA, and transfer RNA, DNA. So mRNA is the one which contains the genetic information. It's the carrier of genetic information for the synthesis of one specific protein. DNA carries gene. It carries actually all the information. Okay. This one stores the information. The carrier of information. The carrier of information is the RNA. So the information is found in DNA. This information found in DNA will now be taken to RNA because RNA is a carrier. And then specifically messenger RNA. Okay, because it has the messages for the synthesis of proteins. And then now you are going to go to proteins. So DNA stores the information, RNA is a carrier. So that is it for this class. If you have loved it, you can get to register with us and revise in mathematics, biology, physics, chemistry, even GME 1115. 0974016268. We have got all topics worked out with revision of exam past papers just like this one. So you do well to prepare with us. Thank you so much and stay ever blessed.